Have you ever wondered what really happens to a drug once it enters the human body? Where does it travel? How does it break down? And what hidden biochemical steps does it go through along the way? To uncover these answers, scientists rely on a powerful and elegant tool known as stable isotope-labeled compounds. These compounds allow us to trace drugs and metabolites with precision, even in incredibly complex biological environments. In this presentation, we'll explore why these labelled compounds are so valuable, particularly in pharmaceutical research and metabolomics, and how they help us literally see the invisible processes of the body. So what exactly are stable isotope-labeled compounds? They're simply molecules in which one or more atoms are replaced with non-radioactive isotopes, such as carbon-13, nitrogen-15, or deuterium. What makes them so useful is that these isotopes behave chemically the same as their standard counterparts, but they can be easily detected using techniques like mass spectrometry, or NMR. This makes them act like a microscopic GPS tag, invisible to the body, but perfectly visible to our analytical instruments. When developing a drug, scientists need to answer some critical questions. How quickly is the drug metabolized? Which pathways are involved? And does the drug form any potentially harmful intermediates during breakdown? Stable isotope-labeled compounds provide crystal clear answers to these questions. They allow researchers to precisely trace the drug's journey through the body and identify risks or inefficiencies early, long before the drug reaches clinical trials. This level of insight isn't just helpful, it's required for regulatory approval and safe dosing strategies. In pharmaceutical research, stable isotope-labeled compounds play a crucial role in studies known as ADME: absorption, distribution, metabolism, and excretion. They also support bioavailability and bioequivalence testing, which helps compare how different drug formulations behave in the body. Additionally, they help identify metabolic byproducts and reveal whether other medications might interfere with a drug's metabolism. Together, these applications shape decisions about safety, dosage, and long-term effects before a drug ever reaches patients. In metabolomics, stable isotope-labeled compounds are used as internal standards. This helps scientists make accurate measurements even when working with complex and variable biological samples. They are also essential in fluxomics, where the goal is to map how molecules move through metabolic pathways. This is especially important when studying cancer metabolism or mitochondrial diseases, situations where small metabolic differences can reveal major biological insights. Without isotope-labeled standards, metabolomics would be like trying to read a map that has been blurred by rain. However, there's a practical challenge. Most compounds researchers want to study are not available in labelled form on the market. And even when they are, the labels may not be located at the positions needed for a specific experiment. Research also often requires very high isotopic purity. So, custom synthesis becomes essential. Tailoring label placement and purity ensures the data we get is clear, reliable, and scientifically meaningful. But custom synthesis is not simple. Stable isotope-labeled precursors are often quite expensive, and multi-step chemical synthesis increases the risk of losing the label during reaction steps. Maintaining isotopic purity adds another layer of complexity. This means synthesis strategies need to be carefully planned to preserve the label precisely where it needs to be. The end goal is to ensure that the labelled compound truly reflects the molecule researchers want to track, without compromise. A high-quality synthesis partner doesn't just make molecules. They help design the best route to make them, efficiently and accurately. They provide analytical verification using tools like LC-MS, GC-MS, and NMR to confirm the compound's structure and isotopic purity. They also supply clear certification documentation for research or regulatory needs. And, importantly, they offer flexible production scales, from milligrams for research all the way to multigram batches for preclinical studies. This ensures scientific reproducibility, which is a cornerstone of trustworthy research. Let's look at a real example. Researchers studying a particular drug used a carbon-13 labelled version to trace its metabolism. They discovered an unexpected intermediate that only formed in patients with liver dysfunction, a metabolic step that had gone unnoticed before. This discovery helped redefine dosage guidelines and prevented potential toxicity. It also highlighted the power of stable isotope-labelled compounds in enabling personalised medicine, where treatment can be adapted to individual patient conditions. Stable isotope-labelled compounds may operate quietly in the background, but they are indispensable in advancing drug discovery and metabolomics research. They bring clarity to complex biological systems, allow us to track molecules with astonishing precision, and support safer, more effective drug development. In science, progress often comes not from dramatic breakthroughs, but from precise tools that reveal what we could not see before. And stable isotope-labelled compounds are exactly that, tools that help us see the invisible.